I will be talking about diet and epigenetic events and cancer prevention. I don't think anybody's necessarily mentioned the definition of epigenetics in this session yet, but it has a varied definition, evolving definition, we'll say. But one that's commonly used is changes in DNA, changes in gene expression, heritable changes in gene expression without changes in DNA sequence. And these heritable changes in gene expression could also be, at certain times and in certain places of the genome, reversible. And that's where the interest, perhaps, with environmental exposures comes into play. In particular, our interest in diet and diet's potential impact on epigenetic mechanisms. And here's a nice figure just showing the two main epigenetic marks that have been spoken about in this session, DNA methylation, CPG on the nucleotide, and histone modifications in the histone tails. And also, these epigenetic marks are certainly implicated in disease, but they also have involvement in development and in regular processing of all things, many things that are going, many cellular processes. So it's a normal occurrence, but it becomes aberrantly regulated in certain diseases. And of course, the disease that I am going to speak about is cancer. And there has been quite a bit of work because cancer is accessible, tumors are accessible, if you will. So they have been interrogated, as well as cells in culture, transformed cells, cancer cells in culture have been interrogated, as well as experimental models have been developed to look at epigenetics in cancer. And some classic hallmarks of cancer are global hypomethylation, and this is associated with chromosomal instability, and then also, which has been found in these cells, of site-specific hypermethylation, which is associated in cancer with tumor suppressor silencing. And then there's also something that's not talked about quite a bit, but it has been observed, is hypomethylation in specific regions that may regulate oncogenes or enhance oncogene expression. And there's also been more recently aberrant epigenetic marks for cancer for the histone modifications. There's a lot of changes there, as well as in some of the non-coding RNAs. And there's an interesting also chromatin factor, many chromatin factors that regulate the chromatin and also are involved in this epigenetic regulation, one of which is the polycomb repressive complex. It's becoming very important in stem cell self-renewal, which has involvement in cancer development. And there's the theories of cancer stem cells, and this chromatin factor appears to play a role there. Epigenetic regulates several different pathways, as I had mentioned earlier. Some of those are listed here. And also some of the enzymes that are involved in epigenetics become dysregulated in cancer as well. And our interest, of course, the hype, if you will, about epigenetics is that it's potentially environmentally malleable through diet, hormones, and other impacts as well. I think Dr. Milagro had a nice figure showing all the different impacts made upon the epigenome. I'm going to concentrate, I pulled out examples of diet, epigenetic events, and also more cancer prevention. So that will be the highlights here, though I have included some on cancer and some changes in tumors and how diet may impact that as well. So here's a figure showing how bioactive food components may influence these four marks, these four epigenetic marks. And there's quite a bit of evidence for diet impacting DNA methylation. It's probably the most historical, if you will, the one-carbon metabolisms. These have been discussed, and I'll discuss a little bit more of that as well. And also histone modifications are probably in second place for the most studied with regard to diet's impact there. And very little research has been done for how diet may impact chromatin factors and its regulation of gene expression, as well as the non-coding RNA. And I really like Dr. Martinez's movie yesterday. I think that as time goes on, it's really difficult to discuss these marks, if you will, epigenetic marks, in isolation, because they all sort of, for certain regions of the genome or a certain promoter, they work in combinations and in sequential order. So in a way, a movie really portrays that a little better than this reductionist approach that I have here. But that's how we'll proceed, and hopefully end up with a little more of the integration near the end. A classic example for the association of diet and cancer is the methyl-deficient diet that induces spontaneous liver tumor. And Dr. Milagro talked about Igor Pogribny's work, and I'll also talk about this. But this was one of the first findings, this study here, and this is in liver cancer. And so choline and folate deficiency resulted, unbeknownst to the researchers at the time that this would happen, in tumors and cancers. This is without a carcinogen, and also what they found were striking changes in epigenetics. And it was thought that these are the defects that are occurring, resulting in the hepatocellular carcinoma and two lung metastases. These alterations are site-specific hypermethylation and hypomethylation, global hypomethylation, and altered DNA methyltransferase activity. Now, Dr. Igor Pogribny has gone on to look at a little more closely. The former slide was in the tumors, so this was after many weeks, 76 weeks. And the next couple slides that I'll show concern more earlier time points to get at the cancer development and what's happening, that maybe we can have some answers for cancer prevention. So he was interested in the significance of DNA methylation, significance or contribution of DNA methylation abnormalities and liver carcinogenesis induced by this methyl-deficient diet. And he was also interested in the next slide I'll show, DNA damage indices, abnormalities in liver carcinogenesis induced by the same diet. And what he did was he looked at early time points. This is at the potentially reversible phase of liver injury to liver hepatocellular carcinoma. And he, at both 9 and 18 weeks after the initial methyl-deficient diet, he re-fed these animals methyl-sufficient diet. And it was found that at 9 weeks, throughout the rest of the experiment, the methylation defects diminished. And also he also looked at a marker for progression for liver cancer, and these were not found. But at the 18-week time point here, he found, even with refeeding of the methyl-sufficient diet, that there was a continued defect in the DNA hypomethylation, and thinking that this contributed to the liver carcinogenesis. And he also correlated this with a marker for liver progression. So it's very interesting. And so this is one believed to be one of the early defects in this model. And here's the results for DNA damage. And here you can see the design where control animals had sufficient methyl-sufficient diet, and then at 9 weeks, up to 9 weeks, they were deficient, switched over to sufficient, and 18 weeks deficient, and then sufficient through the remaining of the study. And at both 9 and 18 weeks of sufficiency, 2 DNA damage markers were reversed, if you will. The DNA damaging effects caused by the methyl-deficient diet after refeeding the methyl-sufficient diet at 9 or 18 weeks was minimized or lost, suggesting that DNA damage is not implicated in the liver carcinogenesis, and the DNA methylation was more important. The story, however, for this association with oxidative stress or DNA damage and DNA methylation perhaps does not end there. There's some evidence for an association between these two mechanisms, if you will. And not too long ago, an observation was made that oxidation of either the single guanine or 8-oxyguanine or of a single 5-methylcytosine to 5-methylhydroxycytosine inhibited the binding of this methyl CpG binding protein that is an important player in silencing of the gene. And so this was thought that oxidative DNA damage could result, therefore, in heritable epigenetic changes in chromatin organization. And of course, we're all interested in this because of the rich evidence for which nutrients and bioactive food components may regulate that oxidative, oxidative balance in the cellular milieu. And also, it's very interesting to note that this 5-hydroxymethylcytosine is becoming a new player as an epigenetic mark, if you will. It's thought that it may be prompting a loss of DNA methylation. For a long time, there was this controversy about whether there was any active DNA methylation. So this is also interesting new work to look out for, and it seems to be an important marker as well, this 5-hydroxymethylcytosine. And I'm going to continue a little bit more on findings in this methyl deficiency diet fed to the rats, because Dr. Pogrebny from the Food and Drug Administration has continued to look at various epigenetic marks in this model. And here he has looked at early changes for some of the histone marks. Some of these histone marks are related to suppression of certain genes, and some are related to activation of certain genes. So he's starting to look at what's happening early and what is really going on. What is the event? Is it DNA methylation? Is it histone methylation? And yet, we don't know the answer. Dr. Pogrebny is also looking at microRNA expression. Now, microRNAs are involved in regulating translation of the messenger RNA. Very interesting work that is happening with cancer and the relationship to microRNA and how that's regulating many different processes. And so he's looking at this more recently, and in the hepatocellular carcinoma, the endpoint, he's found that many changes in the microRNA in these methyl-deficient rats, as shown in this table, and some of the early markers, I can point them out, it's not necessarily on this table, but the first two microRNA, as well as the second to the last one, are thought to be early microRNA expression changes in before the irreversible course to hepatocellular carcinoma. So he's also interrogating microRNA, perhaps, as the initial signal for some of these defects in epigenetics in hepatocellular carcinoma. Though it hasn't been mapped out completely what is happening, what is more important, there's a lot more perhaps to come in this story. I wanted to now switch gears and talk about another interesting model that this was just published recently. So again, I'm trying to hone in on examples about cancer prevention, if you will, or how to change a cell that is in the process towards cancer. And here's a study looking at the effects of glucose restriction on longevity and cancer inhibition through epigenetic regulation. And he chose cells that were of similar origin, a normal cell and a precancerous cell. These were lung fetal fibroblasts, restricted glucose pretty much completely from the cell culture media, and found changes, of course, in cell growth and other markers that would be indicative of energy restriction. And he found that through epigenetic regulation of a couple different genes that are important in cancer, that the normal cell was regulated towards greater longevity in glucose-restricted environment, but the precancerous cell also had a different change in epigenetics with cellular death and cancer inhibition. So very interesting. I think it's one of the first studies in cell culture. It seems very, very simple, but this change in glucose restriction resulting in epigenetic changes in a tumor suppressor gene and a telomerase gene, that both of which are important in cancer, and changes in the phenotype of the cells. I'm going to switch now to some human data, and this is from human epidemiologic findings. Sorry for the slide. There's only one thing that's really important that I'll point out on the slide, and you'll have to trust me for the rest, but we can talk about it after if you'd like. But this is a study that looked at different times of exposure where there might have been a persistent environmental conditions such as caloric restriction or famine in young adulthood. Now this is similar in some of the cohort is the Dutch famine cohort that was spoken about by Dr. Ordovas the other night, as well as Dr. Martinez. But in addition, so this is overall a Netherlands cohort, and they have included in this cohort individuals or subjects who are in the hunger winter cohort and other individuals at other time points. So they teased out 3 different time points for which there may have been caloric restrictions. So they didn't go back and ask these individuals for caloric intake in this study. I think they have that for some of their other studies that they've done. But here, for those who were exposed during adolescence and young adulthood during the hunger winter in the western areas, there was a change in a specific type of associated with epigenetics, a colon cancer. And so this is a CpG island methylator phenotype. It's a sporadic colorectal cancer that is recognized for its CpG island methylator phenotype, because many of the known tumor promoters are methylated and suppressed in these individuals. And so what they found was for those that were exposed to the hunger winter, and they wanted to know whether there was a relationship with this CIMP, the CpG island methylator phenotype. They found that there was a protection, actually, in those who had the more severe restriction in this type of colorectal cancer, suggesting, unlike some of the other things we've heard about the Dutch famine, that it's all bad for you, and probably famine is really bad for you. But for these individuals, there was some protection for this particular type of colorectal cancer. And here is the evidence. It's a 0.65 hazard ratio with a confidence interval that is statistically significant. And going from methyl deficiency, caloric restriction, wanted to talk a little bit about fruit and vegetable consumption and its potential association with epigenetics and cancer prevention. And here's a cohort in which they had information on diet by food frequency questionnaire information, and then they also had samples of sputum from smokers. And in these samples, they were looking for a spectrum of DNA, promoter genes that are regulated in their promoter by methylation, known to be associated with lung cancer. And what they found was that those individuals who had consumed more leafy green vegetables had a lower amount of these markers for hypermethylation in the sputum. So this is like a biomarker study. And they also found for folate also a significant association. Those who consumed more folate compared to those who consumed less, let's say, had a lower DNA methylation in a select panel of these tumor suppressor genes that would be hypermethylated. So fewer of those hypermethylations. And also there was some suggestion for multivitamin use being protective as well from the DNA methylation profile. So another interesting association for cancer prevention potential for fruits and vegetables through a mechanism of epigenetics. And I wanted to have just 2 slides, more or less about cancer prevention and more on cancer, if you will. In this study, they categorized, it was many individuals, about 750 women with breast cancer. They took the tumors and looked at DNA methylation profiles in these tumors, and they categorized the methylation profiles using an unsupervised clustering technique. So these classifications are likely based on more or less methylation of tumor suppressor genes or DNA repair genes, if you will. And what they found for the associations were that these methylation profiles were associated with tumor size, alcohol, and folate, among several other covariates that they looked at. So it's kind of interesting that some of the classes will take folate as an example. You can see the yellow, that increased folate intake was significantly correlated with membership in a specific class, for methylation class, and this was class 6. So just to show you that, we can begin to categorize perhaps different kinds of tumor tissue, people with tumor tissues, and risk factor analysis. And it may be important for risk and also for treatment of these individuals in their disorders. And I have an example from the plant kingdom for, again, not necessarily maybe cancer prevention, but once maybe there is a hypermethylated tumor suppressor gene, our dietary constituents able to demethylate and reprogram the gene for expression, and maybe reprogram the whole cell towards a more differentiated cell. And there's several examples for different constituents in cell culture scenarios. And here's a reactivation of genes by an apple polyphenol extract in a colorectal cancer cell line. And they were looking at a DNA repair gene, and they found after a few days that there was an increased expression of this gene, suggesting, and this is messenger RNA, and here is the methylation, where after, this is the methylation. After the APE, the apple extract was given to the cells, there was less, if you will, less of the methylation in the promoter region of this mismatch repair gene, and it resulted in enhanced messenger RNA expression. So just one example I wanted to show you where there's been quite a bit of work looking at, in cell culture, the ability of different dietary constituents to reprogram, if you will, a tumor suppressor gene or a DNA repair gene. And what we need now are really applications in vivo for this kind of scenarios. Is a real response that is something that we can continue to look at in a prevention or perhaps even a treatment setting. And there are many other constituents from other plants for which there's evidence of epigenetic modifications. And I mostly brought this slide because I just really like it. It's really very colorful, and it shows several of the different types of plants that have either been shown to be involved in DNA methylation or histone modifications. And I'm going to talk a little bit about histone modifications. There's this is a cartoon. It's just very simple. I don't have that video to show you, but here we have just the classic opening of a region of a gene or the genome, which is open, and ability for certain genes to be switched on, and it's signified by lack of DNA methylation and histone acetylation. Now, these are classic signals for opening, and classic symbols for closing, say, the genome on an overall basis, so this is rather simplified, is DNA methylation and histone deacetylation, and that's what the bottom part shows. And I bring that up because I'm going to talk just a little bit about histone deacetylase inhibitors. Now, the histone acetylation deacetylation activities are disturbed in cancer cells, And HDAC inhibitors, there are experimentally used HDAC inhibitors that are not dietary constituents that have been shown to turn on repressed genes, tumor suppressor genes, genes involved in cell growth and apoptosis. And also, it's been shown that these HDAC inhibitors arrest growth, enhance apoptosis, and induce differentiation in several cancer cells. And this one molecule called SAHA, it's a histone deacetylase inhibitor, and it is currently used in some clinical indications for leukemia patients, and it's shown clinical utility as an HDAC inhibitor by closing, if you will, by being an HDAC inhibitor from the previous slide I had shown, that closing the certain gene regions so that they're not expressed. So these may be more like oncogenes, say. So there's been some work in cell culture systems, cancer cell cultures, and in vivo scenarios in animal model experiments showing that this histone modification can be regulated by dietary constituents as well, such as butyrate, which is produced in the gastrointestinal by consumption of fibers, and the garlic constituent diallyl disulfide, as well as sulforaphane. And it's been shown that these constituents all result in apoptosis and also transcription enhancement of some important genes that were otherwise diminished. And this is where the cancer prevention may come in. It has recently been shown in a small human population that this HDAC inhibition occurred after volunteers consumed sulforaphane-rich broccoli sprouts. So they consumed, say, a cup, about 68 grams of these broccoli sprouts, and over a short period of time, you can see that their HDAC activity was diminished after consumption, and along with that, there was enhancement of acetylation in the major histones, histone 3 and histone 4. And these are global indices. So to suggest that after consumption in a normal healthy population, now these are it's a couple individuals, there's some changes in our epigenetics. It's not certain, so it was though found to be a transient effect because it did go back sort of to normal after a day or so. The investigators are continuing to look at this to find out, is this real in a larger population? What's the impact or the effect of this? Is it modulating any gene expression? So they haven't looked at that. And whether, this is a beneficial response. Would, chronic consumption of sulforaphane or similar like vegetables be doing this, or would there be some adaptation over time? These are all questions. So what's happening in the normal situation is of interest, and so then we can perhaps understand different responses in individuals on different diets, and whether there's any implication for cancer prevention. And here is a very recent study that I found, I guess it was published last year, but it's this whole idea of mandatory folic acid fortification is interesting with regard to cancer. And here's just an example looking at a histone mark. This investigator looked at the mark of histone 3 lysine 9 methylation, which is associated in certain cancers for gene silencing. And what they did is they had a small population of about 90 women, and this is in cervical cancer, pre-cervical cancer. This is cervical intraepithelial neoplasia that they were looking at, and so it's a histological indication of progression, if you will, towards cancer. And they had a population of women who had samples on that for which they were not exposed to the mandatory folic acid fortification. And then they had a group of women who were exposed to the mandatory folic acid fortification. So this is all, this is in regards to, in the US, there was mandatory folic acid fortification to grain products in the late 90s. So these investigators were interested, is there any difference before and after this mandatory folate fortification in a histone mark that is perhaps regulated by folate consumption. And so what they found was that histone, this methylation mark on the histone was higher in higher grade lesions compared to lower grade lesions. And so that may make sense because they have found this in other cancers. And that histone 3 lysine 9 methylation was higher in women diagnosed with the higher level of this marker in post-folic acid fortification period compared to pre-folic acid fortification period. But they did not find that opposite in the lower risk group of individuals. In fact, they found this methylation was lower in the post-fortification group. So suggesting, again, I think we have other examples for folic acid that if there's a population that may be initiated in cancer and they get more folic acid, there may be a risk possibility and compared to those who are not initiated. So this just raises an interesting scenario in that regard, and also perhaps an interesting marker for diet and cervical cancer. There's several other histone modifications that have been cataloged by that occur through dietary factors, some of which I've talked about and others have talked about. Will go on. And also, I just wanted to talk about one other the chromatin factor, polycomb repressive complexes, because there's a little bit of evidence for how diet may impact this. So these complexes are involved in repressing, and there are a multitude of components that make up the complex. And one of the components in PCR1 is this BIM1 protein. And a study has been done for which, in cell culture, in squamous cell carcinoma cells, that the polyphenol from T, epigallocatechin gallate, has been shown to mediate this epigenetic regulator. And in fact, the EGCG, the polyphenol treatment, reduced the level of this and also enhanced its phosphorylation. And this was associated with reduced activity and overall reduced activity of this polycomb. And so then these cells went on to become less growth potential and less survival, so associated with a more differentiated cellular profile. I wanted
2 minutes.
I wanted to end, and I took more time than everybody else, sorry about that. I wanted to end with the NIH roadmap epigenomics. And so what I've pretty much talked about is global looks at these marks on the chromatin, as well as specific changes and epigenetic changes. But the NIH roadmap, these are projects that are funded by the National Institutes of Health to look globally at specific marks in many different cells. So they have funded 4 different mapping centers, and they've also funded projects on relationships to different diseases and health and different exposures, as well as new technologies, and also looking for new epigenetic marks. So the story will become perhaps even more complicated. I wanted to mention that just recently in October, the 4 mapping centers made publicly available some of their data from their mapping. They have 300 maps on about 56 different cell types, and that's all available at this link right here. So one can use this information as a reference point for future studies, and more is to come. Now, these cell lines that they used are all human. They're embryonal stem cells or adult stem cells, as well as ex vivo tissue samples that they did look at. And this all fits in, as has been mentioned by the other speakers, of epigenetics being involved in the nutritional epigenetics being involved in trying to understand the variation in response to dietary constituents. Thanks.